Hi, everybody. Welcome to our May 3rd Thursday presentation. We have a new guest, new to us for sure. But first, I just want to remind you that next month, June 11th, we will have an in-person R&D Committee meeting. Kate just told me that the last one was November, which I would have said February. It didn't seem like it was that long ago. But we will again be discussing priorities and the way that we can dovetail with the wine grape industry task force, and which brings me to another point. You probably all would have received an email asking you to fill out a survey for the wine grape industry task force on for the climate resiliency group. And if not, you will have received an email reminder about this session today that also has a link to that survey. So please fill out that survey because it's really useful for the task force, and the results of the survey will be presented and discussed at the June 11th meeting. So have a look at that and please do it. And now I'd like to welcome our guest. Our new bug guy, the entomologist who's been hired at the station to replace retiring Tom Lowry, Dr. Asim Renyard. He is mostly what I want to say is recently he has been the chair of the Equity, Diversity, and Inclusion Committee for the Entomology Society of BC. He completed his BSc from Simon Fraser University, and then his PhD in 2024, also from Simon Fraser. I'm going to let him tell you everything else, because that's what he's here for. Thanks for coming, Asim.
Okay, yeah. Thank you all very much. So yeah, hi everybody. My name's Asim. I'm a research scientist, an entomologist here at Agriculture and Agri-Food Canada, and it's a pleasure to speak with you all today. So since I'm new to the group, I thought I'd give a little more background about who I am. So I'm somewhat familiar with the area. My dad is from Vernon, and so I used to come to the Okanagan as a kid. But I grew up in the Lower Mainland and completed my PhD at SFU in April of 2024. I then moved up here to Summerland earlier this winter. Chat. Oh, sorry. And moved here to Summerland in the winter and joined AAFC in February of 2025. And I'm an entomologist of woody perennial fruit crops. So my mandate sort of covers grapes, but also tree fruits as well. Now, before coming to AAFC, I spent the start of my career studying ants. And while we're already working on our grape cluster projects, they're still in early stages. So I figured since this is our first time meeting today, I'll spend the start of my time to talk about some of my past work, to give you some examples of the type of work I can do in case that sort of inspires any ideas. I also think ants are cool and their roles in agriculture are often underappreciated. So hopefully at the end of today's talk, you'll be more intrigued by ants and the role they might be playing in your own operations. So for the first part of today's talk, as I was saying, I'm going to walk you through some ant examples. And hopefully from each of these examples, you'll also see how this type of work can connect to our management of grapes. And so these examples are trail pheromone identification, vibrational communication, and our development of a new lethal liquid bait for ants. We'll then get into our SCAP updates, which are largely ongoing work, but I'll update you as best as I can. And we'll focus particularly on activity 4.1 and activity 5, which my lab has a role in. So let's start with our ant examples. So ants are one of the most ecologically successful animals. They're highly abundant, occupying nearly every single terrestrial landmass on Earth. They're often referred to as ecosystem engineers, as their presence literally alters the physical, chemical, and biological conditions of their environment. And it's through activities like nest defense, nest construction, brood care, and foraging that ants really affect the world around them. Their nest defense and aggression alters what other insects can coexist in their environment. Their nest construction leads to changes in habitat, as well as nutrient cycling and decomposition cycles. And lastly, their foraging habits has really big effects on the environment around them. Ants are often influential herbivores and predators in many ecosystems. The collective behaviors of ants are so robust that they even work in outer space. So researchers have actually sent ants to the International Space Station, and while they found that some of the behaviors of ants are slowed down, ants were still ultimately able to function even without the presence of gravity. Now, these effects are not just limited to wild systems or outer space. Ants can also affect agriculture. And so a recent global analysis on ants in agriculture showed that ants promote honeydew-producing pests, so they have a positive effect like shown here, but they also reduce non-honeydew-producing pests, which is maybe not that unexpected for most of us here. But on the whole, they have a relatively neutral effect on pest abundance. And additionally, if you look at their effects on the crops, ants actually reduce plant damage and increase plant yield. Ants can also affect other things in farms. Their digging activity can increase water infiltration into soil, and the soils around their nests are also found to be more nutrient rich. And the reason I bring this up in agriculture is because ant diversity and abundance can be affected by our farming practices, which also affects the ecosystem services they provide. And in Canada, there are actually pretty much almost zero studies on ants in our farms and vineyards, or how they might be affecting things, whether it's positive or negative. So perhaps for a future round of funding, we can talk about ants, and I'm not just saying that because they're my favorite type of insect. So because ants are such a powerful force, it also means that we as humans also need to treat them very carefully. These maps in red show the distribution of just 4 invasive ant species. And globally, there are 309 species that have established in non-native environments. The population of invasive ants often explode due to their aggressive foraging behavior, high reproductive capability, and lack of co-evolved predators and competitors. Because of their large populations, they displace most native ant species and other insects. They disrupt pollination and seed dispersal, have negative effects on wildlife, and have been even found to alter entire food webs. Many invasive ants also sting, making them a direct medical concern for humans. And invasive ant populations are often very high in agricultural systems where they can cause significant damages. And so these costs can range in the billions of dollars. And so while ants are fascinating and deserve a lot of appreciation, we as humans also need to treat these animals with more care to avoid more situations like this. So for most of my studies, I focused on 2 species, Western carpenter ant, or Camponotus modoc, which is native to the west coast of North America. It is also a wooden structural pest. And then the European fire ant, Myrmica, which is invasive in the greater Vancouver area and other parts of North America. Each of these ants has their own distinct biology, and we're going to explore some questions using both of these species. And in case you forget which ant I'm talking about, I'll throw this little cartoon up on most of my slides so you know which ant I'm referring to. So let's start with our first project looking at pheromone identification. So ants forage collectively, meaning that they'll recruit nestmates to food sources that they like. Basically how this works is that an ant at a food source will mark a chemical trail back to her nest called trail pheromone. This trail pheromone originates in various pheromone glands in the ant's body, and where it comes from just sort of depends on the species. This pheromone serves as a chemical guideline for her nestmate to find the resources that she fed on. Ants will mark stronger trails in response to resources that they perceive to be better, and weaker trails in response to poor resources. Exiting ants will then follow stronger trails, and over time, this leads to a collective choice by the nest for the best resource. But in ants, there's still relatively few species with identified pheromones, and our taxonomic range is pretty limited. And so we had an objective to identify trail pheromones used by ants. And we had 2 main hypotheses that we're going to explore today. The first is that ants will follow trails of pheromone gland extracts, and then the second is that they'll follow trails of specific pheromone chemicals. And so how we tested this is we took the extracts and applied it to the edge of a filter paper disc, which we marked with 1 centimeter increments. And then we just basically scored the distance followed by the ants. And we can see here in this example, this ant seems to be following this trail pretty well. And so here's our results. So on the y-axis, we have the distance followed by the ants. And then on the x-axis, we have our control and our extract. The gray symbols represent the response of an individual ant, and then the black symbols in the whiskers are means and standard errors. And so what we can see here is that our extract caused significantly more trail following by these ants. And so from our extract, we then took it and analyzed it using gas chromatography mass spectrometry, or GCMS. Now, through a combination of electrophysiology, literature review, and a bunch of experiments I'm not showing here, we had narrowed down the molecule we thought was the trail pheromone in our carpenter ants to this 2,4-dimethyl-5-hexanolide. The issue here is that this hexanolide exists in 8 possible stereoisomers. So this is their 3D configuration. Essentially, in some molecules, carbon atoms can be attached at different angles, leading to multiple 3D variations of the same molecule. Another way to think of this is that each of your hands are basically mirror images of each other. And molecules can behave the same way, where they look similar, but in 3D space are different. Insect receptors are also often specific to certain 3D configurations. And so how can we narrow down this down to figure out which one of these isomers are the ants using? Now, to orientate you to these graphs here, on the y-axis, we have counts, which is basically abundance of these chemicals. And then on the x-axis, we see different peaks. And each of these peaks corresponds to 1 or more chemicals present in our samples. So how can we figure out what isomer is in the ants? So this peak here corresponds to the isomer that's present in our ants. And we compare this to a synthetic mix of all 8 isomers. And fortunately for us, 2 of these isomers separated into individual peaks, one of which lined up with what was in the ants. Now, from the literature, we had a pretty good idea of what isomer we were after. And when we compared this 2S4R5S isomer to our single peak from all 8 and the extract, they all lined up. So without having even done any behavioral tests, we already had a pretty strong evidence from a chemistry perspective of what molecule the ants were using. But to really drive this home, we wanted to put this one isomer to the test behaviorally. So from our GCMS analysis, we were also able to identify all the potential chemicals in our extract, which we formulated into this trail pheromone blend. So this was basically everything in our extract that was relevant in a synthetic mixture, including our hexanolide. We also tested the hexanolide at all 8 possible isomers. And lastly, this single isomer we found to be in the ants. And what we see here is that this one isomer has an equivalent response to these other 2 treatments, indicating that it's responsible for the trail following behavior that we're seeing. And so our takeaway here is that this trail following behavior is mediated by this one specific isomer. And so from this research, we were able to identify a pheromone component for our carpenter ants. And like I said at the beginning, our taxonomic range for this work is pretty limited. And so what was cool is when we tested this in a different genus of ants, we maybe not surprisingly found a whole new class of chemicals that haven't been identified in ants before. And from these more fundamental chemical ecology type studies, I think we can build it into some pretty cool things. And we've now expanded this work beyond identification to see how ants respond to pheromones for species which share the same habitat in their communities. And we found that ants actually do adjust their behavior in response to trail following when in the presence of other ant species pheromones. So we found evidence that supported our 2 hypotheses. And the reason I bring this up as an example is pheromones, as we all are aware, are very useful as a tool in agriculture for lures, for monitoring, mass trapping, or mating disruption. And in fact, we've already begun a collaboration in grapes on this. So we're currently working in collaboration with my former PhD advisor and his wife. So this is Gerhard and Regina Grease, and they're both researchers at Simon Fraser University. And there are reports here and in Ontario that the pheromone lures for grape mealybug are not really effective. And so we're looking into why that is the case and potentially trying to create new lures for growers and also for researchers so they have better tools to monitor for mealybugs. And so this is a collaborative project that we've just started with researchers in Ontario and at SFU. So let's move on to our next example, vibrational communication. So a cool behavior that ants do are these drumming behaviors. And we see here, after I poke this ant with the chopstick, the ant starts rapidly tapping her body against the ground. And this is typically done when ants are distressed to alarm their nestmates. If we slow this down and look at it under high speed, we see that our ant starts in a resting position, raises her body up, and then strikes it back down against the ground. And this generates a substrate-borne vibration, which can then be sensed by her nestmates. And in ants, vibrational communication has rarely been studied in only a handful of species. And many of these studies took place decades ago. What we do know is that these types of signals mainly cause locomotion changes, so ants may stop moving or running faster in response to these types of signals. And so we had a new objective, which was to characterize and behaviorally bioassay vibratory alarm signals in carpenter ants. And we had one main hypothesis, which was that these vibratory alarm signals would cause freezing, rapid running, but not attraction of ants. Before we could do any of this work, though, we had to record and characterize this signal. And we recorded this vibration by putting an ant onto a thin wooden strip. We then used a metal wire attached to a speaker to record the vibration, kind of like a microphone. We also had this laser Doppler vibrometer here to monitor the quality of the recording. And so as the ant is drumming, it's vibrating the wood, which is picked up on the metal wire, which is recorded by the speaker. And so then that just sort of creates an audio file of the ants drumming. And so we did this with several times with several different ants, and we created one giant audio file to use during experiments. And so how we actually tested this is we can use our recording of the ants drumming to impart this vibratory signal onto a thin wooden surface. So we had this ant dance floor bioassay design, where what we did here is we had this plexiglass arena with a suspended thin wooden floor, and that helps with the signal transmission. I've then introduced 5 ants into this arena here using this holding tube. And after all the ants had exited the tube, I began video recording the ants for 5 minutes. And after 5 minutes, I'd begin to play back the vibratory signal. After this, I would continue to video record the ants, so we had a measure of the ants' behavior before and during experiencing the signal. And using these video recordings, we can track things like their freezing behavior and running speed. I also drew these circles around the base of each metal wire on the wood, so we could see how often they would visit the area around each metal wire as a measurement of attraction. And so here's a video to give you an idea of what this looks like. So here we see some ants walking around normally, and then as soon as they experience the stimulus, they all start to run really, really fast, and then they all sort of just freeze. And if you've ever been around ants, ants standing still is pretty uncommon. So it was pretty interesting to see these kinds of behaviors shape up. Now, before I get into our next set of results, I thought I'd show you more detail about how we got there. So we used automated tracking in the software program Fiji to track the movement of the ants. And we used this specifically to determine their running speed in these experiments. Okay, so here's what our results are. So on the y-axis, we have the time spent frozen, so basically the time spent not moving. And we have before and during the vibratory stimulus on the x-axis. Each colored point is an individual replicate, and then the black symbol and whiskers are means and confidence intervals. And we see here that the ants spend much more time frozen and not moving while they're experiencing the vibration. Similarly for running speed, which is here on the y-axis now, we see that the ants are running faster in response to the vibration. In terms of attraction, on the left panel, we see very similar visitation to either one of these stimulus speakers, which makes sense. Both are on but not playing anything. When one speaker is playing, though, we see also a similar response between each speaker, which is indicating to us that this signal is not attractive to the ants. So what does this all mean? Well, reduced attraction and increased freezing could be an adaptive response to avoid predation because many predators use motion to detect prey. So you either want to not move, or if you're going to move, you want to move really fast. And so that gave us evidence to support our hypothesis. And the reason I bring this up is an interesting thing is that many leafhoppers also communicate in a similar fashion, but for mating. And in this case, both males and females call to locate each other on plants. And researchers in California are already looking into this, where they've attached these devices which play female calls onto the metal wires which run along the vine rows. And this signal then gets transmitted through the plants, which actually drastically reduces the number of mating pairs. It's still unclear if it's totally effective for actually reducing plant damage, but this research is is ongoing by folks down in California. Okay, let's get on to our last example, a new lethal liquid bait for ants. Okay, so we've mostly focused on the carpenter ants so far, and now finally we can also talk a little bit more about our European fire ants and how we developed a new bait tactic for these ants. So like I said at the beginning, the carpenter ants are structural pests. They excavate tunnel galleries in wood to create their nests, which weakens the wood's structural integrity. And I also mentioned that our European fire ants are an invasive species. This is their current distribution, which is relatively small compared to other invasive ants. But most pest ants are really difficult to control. Spraying pesticides is often ineffective and has undesirable non-target effects. And to date, there aren't many biological control programs that have been effective for ants. And these ants can drastically alter what biodiversity is present in areas that they invade, and they also reduce property and recreation values. One possible method of pest control that have really good prospects, though, are these poison food baits. I'm sure most of you have seen these in hardware stores. The central idea here is that you have a food bait mixed with an insecticide that the ants will feed on and then bring back to their nest. Once they return to their nest, they engage in food sharing behavior, or trophallaxis, where they orally share food between their nestmates. So this could be worker to worker, worker to larva, or worker to queen. And so this just allows the poison to sort of self-distribute throughout the whole colony. The problem with these baits, though, is many have inconsistent feeding by ants, either through time with the same species, or they don't appeal to multiple species of pest ants, which makes them difficult to market. And so in order to make a bait that has consistent feeding activity, we needed to look at what we can do to improve the appeal of these baits. And the approach that we took was to examine what features do ants look for when they're feeding on food sources in nature. And so the overarching question that we explored was, how do ants select food resources? So just like human foods, ant food resources are basically just complex mixtures of many different macro and micronutrients. And just like us, ants require a balanced diet of these nutrients to survive and function. Two really important ones are carbohydrates, so these are things like sugars, and proteins, which are made up of amino acids. And so we wanted to see if ants evaluate these food resources based off the presence of particular nutrient molecules. So that leads into our objective, which is to identify nutrients used by ants when they select resources. And we'll examine if ants will select resources based off the presence of particular nutrient molecules and exhibit seasonal shifts in nutrient preference. Okay, so let's start off with carbohydrates or sugars. So for these experiments, we used both the carpenter ants and the fire ants. And what we did is we dissolved our nutrients in liquid form and dispensed them in these little Eppendorf tube feeders. We would weigh the tubes in before and after to determine their mass loss. And we also included these evaporation controls to account for passive water evaporation. For the carpenter ants, we used a separate tank connected to their nest. And for the fire ants, we did it directly in their nesting tank. We allowed the ants to forage for several hours before we removed the tubes to determine their final mass. So it's well established that ants and aphids, or really any honeydew-producing insect, have a close mutualistic relationship. Ants keep the aphid colonies clean, protect them from natural enemies, and in return, the ants receive a sugary reward in the form of honeydew. So this video here shows one of our carpenter ants tending a colony of aphids on a Douglas fir tree. And we can see here the ant is collecting honeydew from these aphids. So aphid honeydew is composed of many different sugar molecules. Some are monosaccharides like sucrose, some are disaccharides like sucrose, and some more complex trisaccharides like raffinose. And in the literature, it's shown that some ants prefer these more complex sugar molecules like raffinose, whereas others prefer more simple sugars like sucrose or fructose. And so we systematically explored preferences among sugar molecules with our ants. And so I'll just show you a couple of representative results, in this case, using just the disaccharides. So on the y-axis, we have the proportion consumed by the ants, and on the x-axis, we have 4 different disaccharides. And we can see that both the carpenter ants and the fire ants really seem to prefer the sucrose over these other disaccharide solutions, which is actually a sugar that's pretty common in the environment. Then we compared between different mono, di, and trisaccharides, and we see that the common sugars like fructose and sucrose are basically on par with more complex sugars. And so our takeaway from this is that ants forage on common and complex sugars similarly. Okay, let's move on to our protein or amino acids. So in animals, there are generally 20 amino acids used to build proteins, including for ourselves. In ants, 11 of them are supposedly essential for ant brood development. An essential nutrient for any animal is something that cannot be synthesized in the host animal's body, meaning that they have to obtain it from their diet. And so we wondered if maybe ants were homing in on these essential amino acids when they're foraging. And so we tested all 20 amino acids versus the non-essential amino acids versus water. And we found that the non-essential amino acids were consumed basically at the same level as water. Conversely, the amino acids important for brood development, so the essential ones, were almost on their own equivalent to all 20. We saw a similar pattern in the fire ants with the non-essential amino acids being equivalent to water. And although not significant, the pattern was similar with the essential amino acids being on par with all 20 amino acids. So this was cool to see in the lab, but we wanted to see if field populations of ants would behave the same way. And additionally, we looked at how multiple nutrients might contribute to a feeding response. So here's a shot of me at one of our carpenter ant field sites. And so what we tested was water versus sucrose versus our essential amino acids versus a combination of both. And we ran a new experiment about every 3 weeks on the same ant colonies. For the carpenter ants, we mounted the tubes on trees and conifer forests. And then for the fire ants, we ran experiments in a field and just placed tubes on the ground. So let's start with our carpenter ants. So on the y-axis, we have the proportion consumed, and on the x-axis, we have calendar date, and the results are separated by treatment. And the key takeaway here is that across all dates, the essential amino acids were really what the carpenter ants wanted, and then not surprisingly, the blend containing those amino acids was also really fed on. For the fire ants, we found almost the opposite, where the ants really liked the sucrose instead. But again, also the blend is really consistently fed on. The key takeaway here for both species is that the blend had consistent feeding activity. And so returning to our hypotheses, we found that they did select resources based off the presence of particular nutrient molecules, but they didn't actually exhibit seasonal shifts in nutrient preference, which was actually a little surprising to us. But as I said before, the problem with baits, though, is that many have inconsistent feeding by ants either through time or with multiple pest species of ant. But I just showed you that we have a blend of nutrients that we had tested that had pretty consistent activity through time with 2 different ant species. And so could we pair our nutrient blend with some kind of insecticide to develop a new bait? And this is exactly what we wanted to pursue. And so we wanted to develop an effective and eco-friendly ant bait. And our first 2 objectives were to assess the mortality timeline of our bait formulation and then compare the effect of insecticide dose and formulation on this mortality timeline. And so our setup for this was to create small experimental colonies of 2 fire ant queens and 100 workers. We had control colonies, which provided feeders containing just the nutrient solution, and treatment colonies, which were given feeders containing our lethal bait. The lethal agent that we selected here was boric acid, which we chose due to its relatively good environmental profile. And we basically counted the number of dead workers and queens and replaced the feeders every 2 days. So on the y-axis, we have the proportion of ants surviving, and on the x-axis, we have days of our experiment, and we separately tracked workers and queens. The individual data points are the response of an individual replicate, and then the shaded regions are model predictions and confidence intervals. And so what we see here for both workers and queens, we have a much faster death rate in control. We have a much faster death rate in our lethal bait treated colonies, which makes sense. If you give ants a poison, they will die. But the exciting takeaway here is that we have rapid and complete demise of workers and queens, giving us some indication that this could be a viable bait. Next up, we have dose. So we have most commercially available baits often use 5.4% boric acid, which as a fun fact, I was unable to find any sort of reasoning behind why this dose was chosen. So we compared our bait at 1% versus the 5.4% in purple. The workers here had a slightly slower death rate in the low dose, but the queens were similarly affected regardless of dose, which is really the target here for most baits. We also tested our bait formulated as a liquid or as a gel, and we see that the liquid bait has a much faster death rate than our gel bait. And so our takeaway from this is that the low dose is sufficient and that our liquid is better than the gel. Next, we wanted to evaluate the effectiveness of this bait in field populations of invasive ants. So we took this idea to a field site infested with European fire ants. This field was split into two sections, one control site and one treatment site. The control site received our non-lethal bait, which was basically just the nutrient blend, and then the treatment got our lethal bait. So we set up these little bait stations marked with flags, which were protected by these little plastic lids, and we dispensed our bait in 15 mil falcon tubes. And twice per week, we would use apple baits to monitor the population size of these ants. So on the y-axis, we have the number of ants. On x-axis is our days of our experiment. In blue, we have our non-lethal bait, and in red, we have our lethal bait. And we see here that the two populations of each field site start roughly similarly, with the lethal site being a little bit lower. Then both rise up pretty dramatically, and then only the non-lethal baited site stayed high, whereas the lethal treated site comes back down, telling us that our bait can be viable to reduce ant populations in a field setting. And so we seemingly now have a promising new tactic for an invasive species that we didn't really have any good management options for. The story doesn't quite end there. So last year, or 2 years ago, I was fortunate to collaborate with Dr. Ed Vargo and his technician, Sarah Turner, down at Texas A&M University to run parallel experiments for our European fire ants up here. And then this ant, the red-headed ported fire ant down in Texas, which is a super damaging invasive ant species in the southern US. I don't have any results yet to quite show you yet, but it looks like to have a similar but sort of smaller mitigating effect with these red imported fire ants. And so just to wrap this all up, I hope I've shown you that my research really tries to bring in fundamental biological research and bridge that into sustainable pest management solutions. And I think that our ant bait is a really great example of this. By taking a fundamental understanding of how ants forage, we managed to create a bait that was highly effective for these ants. So that wraps up our examples, and let's maybe talk about some of our projects in grapes. And so to start out, I'd like to say that I'm really much interested in what fundamentally drives the ecology of these agroecosystems and using these understandings to create sustainable and long-term solutions for growers. And so in a similar fashion, I want to bridge this fundamental biological research into sustainable agroecosystem management. And just to introduce you to the team we have, so there's me, the guy who knows nothing, but luckily we have my incredible technician, Andrea, who knows a lot more than I do about grapes and is doing a great job managing all the projects we're on. We also have a couple of summer students, Jay Nelson, and hopefully another student, Michelle Ma, who will hopefully join us in June. So let's start with activity 4.1, vegetation management for long-term productivity and enhanced ecosystem services in Canadian semi-arid vineyards. So my role for this project is for Objective 4 and specifically sub-objective 4.1, which is to quantify non-crop vegetation influence on above and below ground co-benefits and costs in Canadian vineyards, and specifically quantification of pest and beneficial insect abundance in these vineyards. And so this project is led by Dr. Mehdi Sharifi and Dr. Dave Ensing. And so I'll be doing the entomology work and some of the systems that they're working on. So there are two main projects. There's experimentally planted cover crop data, where we're planting cover crops and investigating, among a bunch of other things, how the insect community changes. And the second project is looking at naturally occurring vegetation and the insects that occur there. And so in both cases, we're going to be looking at both beneficial and pest insect abundance. And so if we start with the experimentally planted cover crops, so we have 5 sites that have a randomized block design where there's 3 different cover crop treatments along with the control. And there are sites where Mehdi has planted these treatments and will be monitoring the insect communities in each of these treatments. So in each treatment, twice per summer, we're going to sample for pests by collecting leaves from each treatment. These leaf collections will allow us to look at primarily leaf hoppers, but we're also hoping to see things like mealybugs and also leaf hopper parasitoid rates. And then for just sort of a general sample of beneficial and pest insects, we're going to be placing these sticky card traps in each of these treatments. And so we're currently planning out all of the sites for this, and we'll be starting that up in a couple of weeks. The second part of our project is the natural vegetation observational data. And so this is at 40 sites that are spread out throughout the whole Okanagan Valley. And so for this, Dave and his team are going to be surveying vegetation in the natural habitat, field margins, and in the drive row. And we're going to be placing several sticky car traps along the same transects that they're surveying to identify what insects are present in these different types of vegetation. And again, we're going to be doing 2 sampling periods in each summer. Okay, on to activity 5, managing grapevine viruses and their arthropod vectors. And so I'm part of several objectives here, and this project's led by Jose. And the first is monitoring and mapping of new grape plantings for grape leaf roll virus in relation to their mealybug vectors, and then also looking at roguing of these different vines. The second part is about management of grape mealybug and scale vectors. And then the third is looking at red blotch virus and the seasonal biology of tree hopper vectors. So let's start with this first objective, looking at mapping of mealybug populations. So this, I don't have a huge update on this part yet. So like in some previous mapping studies by Jose shown as an example on the bottom, we're going to be mapping new blocks next to old blocks with known leaf roll virus or mealybug populations. We have a couple of sites lined up so far, but if you have leaf roll with new blocks planted next to it and are open to working with us, please feel free to reach out. We'd love to work with you. And we're going to be starting a lot of this work this fall. Okay, so let's move on to objective 2, management of grape mealybug and scale vectors of leaf roll virus, and specifically looking at horticulture oils and insecticide evaluations. So for these studies, we're going to be testing a couple of products for their effectiveness in managing insect pests in collaboration with our colleague Dave Neal. The first is going to be horticultural oils. And at first we wanted to do this at the field scale, but due to our grapes on site being pulled out, we're going to focus our efforts at smaller scale, so maybe the leaf or greenhouse scale. And to that end, we've already started lab colonies of mealybugs, scale, and leaf hoppers, so we can able to still meet these objectives. But we're still open to working with growers if you're planning to spray these oils and would be open to us measuring some of the insect populations. We'd love to do that. And then the other investigation will be on, in a similar fashion, but just on different types of insecticides. And these were already planned at the leaf and greenhouse scale, so we're still on target there, and we're going to be using our lab populations for that. And we hope to have a more detailed idea of what we're going to be testing soon. But a lot of this work will be done this winter when we're not focused on doing the field work this year. Okay, so let's look into objective 2.2, the modeling of pest development. And so my predecessor, Tom Lowry, has shown this figure in several presentations before about leafhopper peak egg hatch. And this was done in 2022 from here at the Research Center. And in a previous talk to the council, he also mentioned that we need more models and expanded models for this. And that's exactly what we're planning on doing here. So we're planning to expand and have already done more work into more pests at more sites. So we have some data on cottony vine scale and leafhopper from here at the Research Center. We've added a couple of sites for leafhoppers and also for mealybug. And so we're kind of proceeding headfirst into all of these models to get as much data as we can to hopefully give better tools for growers. We're pretty tied up with most of these projects already, but if you have any sites with these types of pests, please let us know, because having more options is always great for these kinds of studies. This is just an example map of how we're doing our mealybug sampling. So at a couple of sites, we're trying a few different sampling methods by stratified random assignment in our rows. So at a given panel, we're either sampling using tape, leaf removal, or bark removal. And most of our success so far has been by removing bark strips, and we're counting the number of egg masses, crawlers, and males and females from these bark samples. And so I don't have any, I guess, tabulated results to show you yet, but here's some photos of what we're seeing from the field just from the last couple of weeks. And so here under the bark, we have some early instar crawlers under the bark. This is just a close-up to just sort of show you them moving around, maybe making some new cottony egg masses. Last week, we started seeing lots of male pupa. So here's one emerging from its pupal case. And so he's just kind of trying to back out from his pupal case to get out and fly around, basically. We also have a male here crawling around just to give you a better idea of what the males look like. And so they're very, very small, and they're difficult to see. Here's a photo of a male under the bark from just earlier this week that our undergrad student spotted. And so you can see they're very, very tiny and hard to see. Also from earlier this week, we don't often see things on our tape samples. It seems the bark is the most promising. But here we have some females that were caught, and they actually ended up laying their egg masses underneath the tape. And so it suggests that they're starting sort of another generation of mealybugs. And so far, we seem to be getting really good data for our mealybugs. And we're hopefully going to be able to give growers some, I guess, more tabulated results as soon as we can. This last objective, I don't have a huge update for, but we plan on finding some sites to doing some sampling to see if we can get a handle on what this vector might be for red blotch virus. So we're starting to look for sites. And again, we're going to be mainly doing this work in the fall. So if you have sites with red blotch, please do reach out to us. So yeah, just some final thoughts. If you have older blocks with either leaf roll or red blotch next to new plantings, please reach out to me or Jose. That'd be really helpful for us. And if you have leafhoppers, mealybug scale, or really anything interesting from an entomology perspective, I'm always happy to chat with people. So please feel free to reach out. And I'd just like to thank the Wine Grape Council, our funders, and of course all of you for listening, and our wonderful technicians and students here. And I'll happily take any of your questions.
Thank you, Asim. Yeah, quick questions. You can just turn on your camera or pop it in the chat.
I'll come out of shared screen.
Here's a warning. If we don't get questions, we're going to be talking about my house ants. So
Hi, I was just wondering for the bait trial for the ants, how does it compare to like other commercially available baits? Like, is the chemical composition different or like nutritionally?
Yeah, so most commercially available baits don't often list their ingredients because they like want to keep that information proprietary. But one of the like leading baits, the taro liquid bait, that's usually like one of the better sellers. As far as we know, it's basically just sugar in that bait. And so basically by having like a slightly diversified nutritional profile, you just kind of hit at like what appeals to different kinds of ants basically. So yeah,
Well,
that's kind of the main difference.
and has there been any research like testing it against other types of baits or just the control trials?
Yeah, so we did a follow-up study that I didn't talk about here because we're still finalizing the results from that. It's a little hard to compare, but basically in some trials, our bait performed better than baits that are already on the market. But then in other ones, in lab trials, some of the commercial baits performed better in the sense that they caused quicker death. But in those cases, oftentimes the insecticide dose was really, really high. And so in a controlled like lab environment where the ants can't access anything else,
Right, yeah.
it's sort of expected that they're going to die really, really quickly. But in other trials that other people have done, those same baits in a field setting often perform really poorly because sort of the sweet spot you want to hit with ant baits is that you want something to kill like not too slowly, but slow enough that it has time to sort of percolate throughout the whole nest.
Yeah.
And so in field trials with those other baits, they don't perform very well. And so it's a bit of a challenge to compare across them, but yeah.
Okay, that's interesting. Cool.
Yeah.
Thank you.
Thank you.
So are the ants recognizing that poison is being brought into the nest?
As far as we know, they don't. At least like directly like taste the poison, I guess. Because in our trials, we actually gave them a choice between so in the control, they had 2 tubes, both of which were not containing insecticide. But in the treatment ones, they actually had a choice between a like a clean bait and an insecticide treated one. And so we did that because we wanted to see if they'll start to avoid it over time, and they didn't. That said, though, like ants will recognize like if a lot of their nestmates start to die in a given area, they'll just start avoiding that area entirely, but it's not clear if it's because of the food resource, or if they're just sensing like a whole bunch of my like comrades are dying here. Maybe I don't want to go here.
Yeah, I mean, that's my strategy with the odorous house ants, is that if I kill some of them, the others no, because they give off the odor, right?
Yeah, totally.
Hans, did you have something?
Yeah, thanks. Can you hear me?
Yeah,
Yes.
can't see you though.
Yeah, I don't know what's going on with the camera. Maybe it's slow. Well,
I know what you look like.
yeah, it doesn't matter, right? Yeah, thanks very much for the presentation. It's really fascinating. So two separate questions. One on the ants. It's really fascinating to see the level of communication within an ant colony. But I mean, you seem to basically, I mean, right now you have tracked more the tracking system that they use to follow each other. How much more subtle does that actually can that be in terms of what they communicate to each other? Because I'm really fascinated by how organized the ant colonies are. I mean, it is almost like a colony makes up one brain, and there must be a lot of very intricate communication going on between all the individual cells in that sense, right?
Yeah, totally. I mean, so I think in general, like we've sort of just barely started to scratch the surface of ant communication, even though it's been, you know, an active area of research for probably like 100 years now. So I presented basically on two different modalities of communication, chemical and vibration. I didn't actually talk about vision or like tactile, like touch type of communication. And even within chemical, I talked about like one type of pheromone produced in one type of pheromone gland in ants. And in most ant species, there are 39 or so different pheromone glands that sometimes produce like multiple messages. And so the way that they communicate, I think, is very, very intricate. But I think it's a lot of it is based off of like simple rules that any individual can follow, where say, for example, with the trail following, it's basically mark a stronger trail if you see the resource is better. And then if you encounter a trail, you follow the stronger trail. And so that's how they like seemingly make this decentralized but collective choice. And I think a lot of their decisions are made like that. So if they're like responding to a threat, it's like, how big is the signal I'm perceiving? And in sort of internalizing their own like physiology and then responding. Does that sort of answer your question?
Yeah, thanks. And I mean, the other one is on the grape leafhopper, the Virginia creeper leafhopper side.
Yeah.
I mean, there is no shortage of these in all organic vineyards in the valley.
Yes.
And you know, I find it interesting because we've been doing this for many, many years, and sort of observing the populations of potential control organisms. So we do have, I mean, the anagris is definitely one with a lot of potential, but we also have a lot of earwigs in the vineyard that do actually eat a lot of eggs off the underside of the leaf. But the interesting correlation between these is that the earwig does not differentiate between parasitized or non-parasitized eggs. So a lot of they actually do reduce the anagris population quite considerably while they are reducing and that is kind of interesting from a, you know, ecological point of view in my mind. But
yeah, that's very
Andrea probably, I mean, Andrea has been on our side multiple times. I mean, she probably knows that quite well.
Yeah, we'll have to chat about that. That's a really cool interaction. It's the kind of thing I like to work on. So that's exciting.
I see you taking notes there.
Yes, yeah, I like to take notes.
Amber is asking if there's a possibility of using the tapping warning sound as a technique of repelling ants, perhaps more for building protection than vineyard versus like chemicals in buildings.
Hard to say. I think in theory, like it could disrupt them enough that ants are often the type that they're not afraid to sort of like leave their nest. And so if something is disrupting them enough, they'll just pack up and go, and almost like move their entire colony. So like in theory, I could see it having some potential. The main, I guess, crux always with any of these types of things is like commercialization and like manufacturing costs and that sort of thing, which is one of the things that's I'm really curious about that research going on with the leafhoppers in California, because that device seems really cool. It's like totally non-chemical. You just hang it up and sort of leave it. But yeah, it would take like, I guess, sort of like an industrial partner to pick that up and manufacture it and that sort of thing.
So my odorous house ant colony,
Yep.
which I've had for years, because I live in the woods and I have a lot of my wood in my house, like open beams and such. But they've left. This winter was the first winter I didn't have this like huge migration across the ceiling and et cetera, et cetera. And I wonder, do you think it's the cold snap that we had last January? Like, are ants threatened that way?
Could be. I mean, they definitely have like lower thermal limits. And so, and like I said, like ants are not afraid to just like pack up and leave. And so they often move. Many ants actually like, they don't actually have like one central nest. They'll have like a single queen, but they'll actually have like multiple nest sites that they nest in. And so it could be that like in your house, you have like one nest site, you know, in your ceiling or wall or something. And then there's like a couple more outside or like, you know, one's in your attic or something. And so they could have just been migrating to like a different part. But it definitely could be the cold. Like if they don't like the environmental conditions, they'll just leave.
Good.
Yeah, which is actually why most people have like, I'm not so sure about here, just because it's drier here, but many ant species in temperate areas really like high humidity. And so oftentimes when people have a house infestation, it's usually actually due to another problem, which is usually like their roof is leaking or they have like a leaking pipe somewhere, and that creates like this humid habitat, and then the ants move in. And so if you are seeing ants in your house, it's also worth the odorous house ant I'm not so concerned about, but if you were seeing like carpenter ants in your house, that's usually like a sign of something more wrong, I guess, in the house other than just the ants.
Yeah, I'm pretty sure that was it's funny when they're migrating because you really can't tell which way they're going because an equal number of ants are moving one way as the other way. So it's
totally.
Yeah. So and I've been told that we in our vineyards, like the vineyards in the South Okanagan that are very sandy, have high populations of ants. And they're, you know, they have interactions with aphids there and that sort of thing. Whereas I'm in Naramata with heavier soils, we don't see them. Or is there any research around that, or is it significant in any way?
I mean, ants definitely pay attention to like habitat and microclimate conditions. And so like in many of like the field sites that I've worked in, like within the same sort of like grass field, you'll see like more ants like closer to the edge of the forest rather than like near the edge habitat. And so like they definitely pay attention to these types of things. And so in that case, it might just be like soil conditions and potentially a couple other environmental variables. So if it's sandier, it might just be easier for them to dig through and create nests. And so that could explain what's going on with them in the southern parts of the Okanagan.
In the photo of you at the collection sites, you were wearing a mask.
Yes, so that was partially to do because that experiment was done during COVID, and we needed to mask up when we had multiple people around. But then also, like when we were outside, it wasn't so much of a concern. But we ants are really sensitive to like if you're breathing on them.
That's
And
what I
so,
wondered. Yeah, yeah.
yeah. So we like it was kind of handy to be able to set up the experiment with a mask on, so you didn't like upset
Yeah.
them.
Yeah, no, that's I wondered if it was something like that.
Yeah.
We have a couple more minutes. Anybody else with a question? You can ask about insect pests in your home, because I did.
Yep.
Asim, did you're probably too young, but did you have a creepy crawly maker? Do you know what it is?
Yeah, I remember those. I'm trying to remember if I had one or if I just like saw them on TV or something.
Yeah,
I feel like I might have. I'll ask my parents
right?
about it.
They should bring those back, man. Because
Those were great.
some of your photos look, very familiar that way.
Yep.
Well, thanks. Oh, Hans, I think.
Yeah, well, since we're talking on about ants in the house, we used in
Yes.
our old house, we used to have every spring we had ants move into the house, and in the fall they
Okay.
would move out. But one year they came in huge numbers, and most of them did go into the aquarium
Oh,
on the water, first floating on the water, and more and more and more would add to this. So in the end, there was an island of thousands and thousands of ants swimming in the aquarium. Do you have any idea what they were doing?
Um,
I've had that in my tea kettle.
Oh yeah?
yeah.
Yeah.
I mean, so one of the things that like ants will do is they can get in like sort of positive feedback loops where because they have these like very defined rules of how they behave. And so like one example of this is I think they're called death spirals. And so if you look them up on the internet, it's basically where ants start following the ant in front of them, and then they form a circle, and then everybody's following the ant in front of them, not knowing who the leader is, and they just walk in a circle and a circle and circle until they all die. And so it could be that something was triggering them to migrate, and they were so focused on migrating and like following those rules that they just like weren't paying attention to where they were going. And so in the springtime, like some ants will have like their mating flights. And so if you were seeing winged ones, they might have been like migrating to get rid of all of the new queens and male ants that are about to take flight and mate. And so they might have been like preoccupied with that, and then accidentally wound up in your aquarium. Ants are funny. They're like, to me, they're like some of the most sophisticated creatures, but then sometimes they also do like very, very silly things, I guess I'll put it.
Yeah, it was very strange. Yeah, thanks.
Yeah, of course.
I talked to a grower who's already seen leafhopper. Does that seem early to you?
I mean, in some of our sites, like we're already starting to see some egg laying, and so it doesn't seem horribly early.
Yeah.
But yeah, I guess it depends on where they are. Like if they're further south, like maybe that would also line up. But
Yeah. Okay, next time we won't ask you about ants. We'll ask you about
I mean,
mealybugs and leafhoppers.
ants are my favorite insect, so I'm always happy to talk about ants.
Thanks so much for coming. And
yeah, thank you for having me.
yeah, we'll be watching to see how the research develops, and I'm sure we'll see you soon.
Great.
And just a reminder to everyone, Kate has put the links to the survey and to register for the in-person June 11th in the chat. And so I think maybe once we stop the recording, we'll just leave it for a couple more minutes if you need to click on that. Thanks again, Asim. We'll see you again. And,
awesome. Thank you all very much. Enjoy the rest of your day.
Yeah, thanks. Bye, everybody.